Welcome again. As we discussed in this video, we are going to solve the second part of the 2021 high-level biology final exam paper. And in the first video, we solved the first, second, third, fourth, and fifth exercise. So today is the time to continue with the sixth one. Intestinal hormones. In the figure, a section of the alimentary canal and the effects of hormones involved in its regulation are shown. Identify the organs marked by the letters, write their names on the dotted line. So it's very easy to identify that this is here stomach, esophagus, stomach, okay, obviously it is labeled, so that is extra help. We have the liver, that cannot be seen in here, but the gallbladder we see. We also know that the pancreas is attached to the duodenum, which is the first part of the small intestine, and basically we named all of the parts that must be named in here. So X is indicating the gallbladder, which is storing bile for us. Y is showing us here the pancreas, and then Z is the duodenum, first part of the small intestine. The excitatory effect of the hormones produced by the stomach and intestinal wall is indicated by plus, while their inhibitory effect is indicated by minus. The hormone gastrin is produced by the wall of the stomach and duodenum in response to food containing amino acids. This hormone increases the secretion of pepsin and hyaluronic acid, gastric juice, to help the digestion of proteins. Based on the diagram, discuss the effects of other intestinal hormones. Fill in the gaps of the following text with the appropriate words. In response to the partially digested food chyme leaving the stomach, cholecystokinin is produced, which induces the secretion of two digestive juices. So let's observe this image. Cholecystokinin is here and it induces bile. And the arrow is also pointing towards the pancreas, so the pancreatic juice. Due to sodium bicarbonate, the pH of these digestive juices is alkali. The next intestinal hormone, secretin, which is here. Secretin induces the production of pancreatic juice, you can see it with a plus sign, and it is inhibiting, so it inhibits the secretion of the hormone gastrin. The hormones GIP and VIP, here you can see them, namely vasoactivate intestinal peptide and gastric inhibitory peptide, you don't have to know them. You can see that it is negatively affecting the hyaluronic acid production, so both prevent too much juice with acidic pH from being released into the stomach. The hormonal effect is supplemented by the excitatory effect of the autonomic nervous system. The taste of the food inherently triggers the secretion of juices by the glands. This response was named unconditional reflex by Pavlov. Exercise 7, multiple aspect classification. Write the number of the species or groups of species into the appropriate places in the diagram. Number 1, decomposing bacteria. They are prokaryotes, unicellular organisms, decomposers, and they are not fungal species. So number 1 will belong here, obviously heterotrophs. So number 1 should be put in here. Panicillium molds, fungal species, and they are decomposers, heterotrophs, and eukaryotes. So number 2 will belong to this box. Mucor species, pig mold on bread, exactly the same characteristical features can be said about them, like about Panicillium molds, so number 3 comes here. Paramecium, which is a unicellular eukaryote, it feeds on organic remains. It's unicellular eukaryotic organism and it is a decomposer, so 4 comes here because it's not a fungus. 5, baker's yeast, it's a fungus but a unicellular one. It's decomposer, heterotroph, and eukaryotic, so number 5 needs to be put in this little box. Number 6, death cap, which is a saprotrophic mushroom. It means that it feeds on non-living organic matter. It's again a fungal species. Decomposers, heterotrophs, eukaryotes, so the same box. Number 7, onion, which is a plant. Obviously eukaryotic cells are building it up. and it's autotroph, so it must be written outside of this big box. So number 7 will come here. Cellulose digesting bacteria, which is basically, again, these are prokaryotic cells, unicellular ones, decomposers, heterotroph. So number 8 must be written next to 1 here. Number 9, humans, eukaryotic organisms and heterotrophs. So I just put humans here. 10, disease-causing agent of syphilis. Prokaryotic, heterotroph, unicellular. So it is going to be in this part. 11, Crowcoccus species, which are autotrophic unicellular bacterium. So number 11 will be written in here. And then earthworms, 12, feeds on organic remains. So they are eukaryotes, heterotrophs, decomposers, but they are not fungal species, so 12 should be put in this place. That's it. Exercise 8, sexes and chromosomes. The mechanism of sex determination is remarkably diverse in the living world. Because of the way the sexes are determined in humans, the sex-linked characteristics are not necessarily transmitted in equal proportions from one parent to the male and female children. Let us suppose that a rare allele located on the X chromosome occur only in one copy in the genome of one of the parents. What are the chances of this allele being passed from the father to his daughter? Father can only give one X chromosome to the daughter, so it's 100%. From mother to daughter, mother has 2 X's, so actually 50%. And from the mother to her son, it's again 50%. As long as the allele in the example above is recessive, it will only show up in the phenotype of men, but not in that of women. Give your reason for the difference. Somatic trait is coded by a single copy of the genes in man. So the effect of one recessive allele will show up in the phenotype. With other words, we can say that men are hemizygous for the trait, so the recessive allele is expressed phenotypically. Let's move on. In birds, the mechanism of sex determination is the opposite of what is found in mammals. The female hen bird produces 2 types of egg cells, X and Y types. Sometimes we indicate them with Z and W, while the male's cocks give rise only to one type, X type, of sex cells. Compare the mechanism of inheritance of the 2 groups of animals. Let us suppose that the Y chromosome does not carry somatic information, other than those required for sex determination and reproductive functions. So, the options can be seen in here, A, B, C, and D, and let's look at the statements. If the sex cells are involved in fertilization in equal frequencies, the proportion of the 2 sexes will be approximately equal in the offspring. The good answer in here will be C. The female partner passes its somatic traits linked to her X chromosome to both her female and male offspring. That's going to be true for mammals. The sex of the offspring is determined by which egg cell is fertilized. It is written down in the text in here, basically, that in birds the mechanism of sex determination is the opposite, so it means that the good answer here will be A. The sex of the offspring is determined by which sperm cell fertilizes, obviously mammals, and the recessive traits linked to the X chromosomes can only affect the males, which is going to be D, so it is not true for them. In the case of some social insects, sex determination is linked to the ploidy level. The diploid individuals are females, the queen and the workers, while the males are haploid. Males, drone, develop directly from the egg cells laid by the queen without being fertilized. That's an important piece of information. Females, however, develop from the fertilized egg cell laid by the queen. The workers no longer take part in reproduction. So you can see everything in the picture. The degree of similarity of genes among different individuals living in one social colony depends on the measure of relatedness among the individuals. In the task below, the term genetic relatedness is used to refer to the probability of a rare allele being present in two different individuals, that is, the percentage of their genome they have in common. Compare the measure of genetic relatedness existing between different individuals belonging to the same social colony. Write the appropriate signs, smaller than, equal, or larger than, in the boxes. It is assumed that the colony is the descendant of one single queen and one male. So, the probability that a rare allele present in the queen is inherited by its male offspring, or the probability that a rare allele present in the queen is inherited by a worker of that colony. So the chances are equal to pass on that rare allele to a worker or to a male offspring. Therefore, I need to write in here an equal sign. The genetic relatedness between a worker and its brother, or the genetic relatedness between two workers. At this point, it's useful to jump back for a second and remember what you read in this text about the males and the females, and if you know how do they develop, actually it's easy to answer the question that the genetic relatedness between two workers will be larger than the genetic relatedness between between the worker and its brother. Okay? In the population of the ant genus Pogonomyrmex, the inheritance of sexes is similar to the previous species, but there are two types of males and two types of females in the colony. This trait is genetically determined and codominantly inherited, which differ in their color as well. Interestingly, workers can only be produced by the fusion of gametes originating from two individuals with different colors, that is a red male mating with a black queen, while a queen can only originate from mating between individuals of the same color. The queen of this species can mate with several males before establishing a colony, but no mating can take place afterwards. The queen lays a huge number of eggs during her long lifespan, thereby maintaining the colony. Although the males are only required for the next generation, the activity of the workers are indispensable. What kind of individuals are produced as a result of a black male mating with a homozygous red queen in the hive? Luckily, we have everything written down in the text that we need to know to answer this question. First of all, it is written down in here that the workers can only be produced by the fusion of gametes originating from two individuals with different colors. So definitely workers will be produced. But as it is stated at the beginning of this text that the population is similar to the previously mentioned species, let's remember what we learned about them. Males develop directly from the egg cells. So obviously males will also be present. And due to the fact that it is a homozygous, obviously red queen, so the males will be red. So the good answer must be this one, B. I hope it was understandable. Okay. What is the precondition for a hive being permanently viable? It should be established by the mating between a queen and a male, both of the same color. If this would happen, no workers would be produced, as we learned from the text that workers can only be produced by the fusion of gametes from two different individuals with different colors. So A cannot be the good answer. It should be established by the mating between red male and the black worker. And where would be the queen? So it is again not good. The queen should mate with several males. Here the important thing is that different colors should be present, and it is not mentioned, so it cannot be the good answer. It should be established by a queen mating with a male of different color. That is going to be the correct answer, and one male should mate with several queens. Obviously, this does not make sense. So, D is going to be the correct answer. What is the precondition for both fertile males and young queens developing in a viable hive? Again, this is a question for which you can find the answer clearly stated in the text, where here exactly the queen of this species can mate with several males before establishing a colony, but no mating can take place afterwards. And actually D is describing the same thing for us, so D is the good answer. It should be established by mating between queen and a male, both of the same color. That's not good, we discussed. It should be established by a queen mating with a male of different colors, with several males. So again, that's not good. The queen should mate with several males where the males are all of the same color. Problem is here that the color shouldn't be the same. And then there is no such hive where both males and females. Okay, that's again not true. So D is the good answer. Okay, let's move on. Exercise 9, an optional task. I thought that I will work out both of the tasks, but you have to know that in real life, when you will do your final examination, only one task should be done. And yeah, that's it. The health of kidneys, polycystic kidney disease. The adult polycystic kidney disease is a heritable disease, the symptoms of which appear after puberty, but normally it is not until the age of 40 that it starts causing problems. Symptoms include urinary tract infection involving a painful burning sensation when urinating, and patients often feel that they are unable to empty their bladder. Kidney stones can also frequently appear along with severe pain in the kidney region, high temperature, and shivering. Bleeding cysts can also cause moderate anemia. In the most severe cases, the symptoms of kidney failure can develop, including tiredness, headache, decreasing concentration ability, lack of appetite, and weight loss. The doctor recommends the patient to drink at least 3 liters of water a day, if not more. Give your reason why, based on the text. As we can read it in the text, kidney stones can also frequently appear. By drinking a lot of water, actually the kidney stone formation will be prevented, so that's what we need to write down. It can reduce the risk of forming kidney stones. Which one of the following medical examinations is not suitable for recognizing the symptoms of the disease? Write the letter of the correct answer. If you look at the options, you can easily figure out that the electroencephalogram is the one which cannot be used for kidney examinations, so D is going to be the correct answer. Ultrasound examination is a kind of imaging test with which you can look at the kidneys. Doctors can detect many diseases simply by looking at urine tests and blood tests, and computed tomography is also used for kidney examination, so D is the good answer. It is observing our brain. The inheritance of adult type polycystic kidney disease is shown in the family tree below. The affected individuals are marked with the black symbols. Let us suppose that no new mutation occurs. Okay, so here we have the family tree. Study the family tree carefully and answer the questions. Is the disease caused by the dominant or recessive allele? Give your reason for your answer based on the diagram. If we look at the family tree, we can see here a very interesting phenomenon. So, if it inherited recessively, no healthy kid would be present. Why can we state that? Just imagine the scenario. If the disease would be inherited like that, it would mean that all of the offsprings would be diseased. But in here, we have two parents who have both healthy and diseased offsprings. In that way, their genotypes could only be the following, okay? So the answer is two diseased parents have a healthy child, so the disease is inherited dominantly. If you want, you can indicate that the two parents we're talking about is 1 per 1 and 1 per 2, okay? Is it possible that the allele causing the disease is linked to the X chromosome? Give your reason for your answer based on the family tree. Okay, so what the exercise is asking us is the following. So we would have this scenario, and if you complete this Punnett square, you can easily see that no healthy daughter would be present. But if we observe this family tree, we can see that here we have a healthy daughter. So the answer is no, the disease is autosomal, not X-linked, because we have a healthy daughter in here, in the second generation. Is it possible that the allele causing the disease is linked to the Y chromosome? Give your reason for your answer based on the family tree diagram. It cannot be Y-linked due to the fact we have a healthy son in here, in the second generation, plus there are diseased females. So the answer is very easy. It is not possible. If it was Y-linked, no diseased woman would be present, and the fifth man in the second generation will also be diseased. Give the genotype of person 1 and 2 in generation 1, mark the dominant allele with capital A and the recessive allele with lowercase a. So actually we have already done that. These individuals will be heterozygous. Person 2 per 1 and 2 per 2 decided to visit a genetic counselor to find out about their chances of having a healthy child. The woman has a healthy daughter from her previous marriage who is not shown in the family tree. What was the answer they received? Support your answer with appropriate calculations. So let's go back for a second to the family tree. So we are talking about these individuals now, and what we know is that this woman has a healthy child. It means that she must be heterozygous for the trait, and due to the fact he is healthy, he must be homozygous recessive. So that is going to be the scenario. So it's a simple calculation basically. Mom will be this and dad will be this. So let's construct the Punnett square. Very easy one. You don't even need to write down twice the A because these are going to be the same. If you write it down then. Anyhow, so there is 50% chance that the child will be healthy. Okay. In Hungary, according to some surveys, 1 out of 200 people is diagnosed with the disease. Let us suppose that the Hungarian population is in genetic equilibrium. Calculate the frequency of the alleles. Give the results to 3 decimal places. So finally, a nice Hardy-Weinberg equation exercise. Don't forget what we discussed. So p squared plus 2pq plus 2q squared equals 1. And another thing you also need to remember is that p plus q equals 1, where this is the frequency of the dominant individuals. So in this case, big 2pq is showing the frequency of the heterozygous genotype, like that, and q square is showing the frequency of the homozygous recessive individuals, like this. The exercise is actually asking us to calculate the frequency of the alleles, and we know that the alleles are actually going to be P and Q. So P is the frequency of the dominant allele, and Q is showing the frequency of the recessive alleles. So these are the things that we need to figure out. So let's see what the exercise is telling us. 1 out of 200 people is diagnosed with the disease, which means that they belong to this or this category. I think it's an easier approach if you start with the q square number, because we know that q square is going to be 199 divided by 200, which will give you 995,000. Okay, and from this number you can easily calculate, if you take the square root of this number, you can easily calculate q, which is going to be 997,000. Okay, so the first number, we have the first number, so the frequency of the recessive allele, and then using this you can easily come up with P simply by doing this 1 minus 0.997, which will give you 0.003, and these are going to be the correct answers. We gave 3 decimal places, so everything is fine. This is the, and obviously you need to write it down, that this is the recessive allele frequency and this is the dominant allele frequency. And finally, the essay type question, urine formation. Write an essay about how the urine is formed in the kidneys of a healthy person. In your essay, include the following points. What are the parts of a nephron? What is filtrate? How and in which part of a nephron is the filtrate produced? In which processes and in what way the water and glucose content of the urine is changed? And which hypothalamic hormone and in what way causes the composition of the urine to change as a result of thirst? So I thought this essay type question deserves a separate slide. So let's quickly revise everything we have learned about kidneys and nephrons in general. We know that the kidneys are filtering our blood, and basically they are helping us to get rid of metabolic wastes plus many other substances. And we excrete all of these substances in the form of urine. We also know that every single, we have 2 kidneys, so the 2 kidneys will contain millions of nephrons. These are the functioning units of the kidney. And basically we are going to look at the first question now, which is about the parts of a nephron. So we know that the afferent artery will enter to the kidneys, and that will branch into many, many arterioles. And finally, the glomerulus is going to be the connecting point. So glomerulus is made up of many capillaries, and this is the point where actually the primary filtrate will be formed. So glomerulus will be surrounded by the Bowman's capsule, or sometimes we can call it a glomerular capsule. These two things together will build up the Malpighian corpuscle. So that's the first thing that must be written down. The first part of the nephron, mass of capillaries, glomerulus, and the Bowman's capsule make up the Malpighian body. That's the first part. Let's move on. After that, we discussed there are two convoluted parts, the proximal convoluted tubule and the distant convoluted tubule, and these convoluted tubules are connected by this U-shaped part, which is called the loop of Henle. And finally, we have the collecting duct, obviously. So after the Bowman's capsule, the following parts are building up the nephron, proximal convoluted tubule, loop of Henle, and the distal convoluted tubule. The collecting ducts will open into the renal pelvis. You can see that many, many nephrons will actually connect to this collecting duct. Okay, second question. What is filtrate? How and in which part of the nephron is the filtrate produced? So filtration is happening right at the beginning of a nephron, where the hydrostatic pressure inside the glomerulus is much higher than in the glomerular capsule or Bowman's capsule. Therefore, plasma with many materials will enter to the Bowman's capsule throughout the fenestrated capillary membranes, and the primary filtrate will be produced here. What is present in the primary filtrate? Water, glucose, many ions, amino acids, uric acid, carbamide, so excretory products. Very important, only small molecules. Large plasma proteins and red blood cells will never enter to the primary filtrate in a healthy person. Okay, so that's what I wrote down in here. Urine forms through three processes. That's what we discussed according to our book, filtration, reabsorption, excretion. Filtration happens in the Malpighian body, so here, where many materials will move from the glomerulus into the Bowman's capsule due to the pressure differences. So in the glomerulus, the pressure is much higher than in the Bowman's capsule, and the primary filtrate doesn't contain red blood cells and large proteins. Okay. Next question, in which processes and in what way the water and glucose content of the urine is changed? So one thing you need to remember is that in the primary filtrate we have a lot of good and useful materials that we don't want to excrete, and you can see this peritubular capillary network which is around the nephron. These capillaries will help us to accept the reabsorbed materials. So what is going to happen is that while the filtrate is moving down these tubes, actually the majority of the materials will be reabsorbed. Okay, so water reabsorption takes place passively, while glucose reabsorption will be an active process, and it's very important that the whole glucose content of the filtrate must be reabsorbed in a healthy person, because if there is glucose in the urine of a person, it is an indication of diabetes. Okay, which hypothalamic hormone and in what way causes the composition of the urine to change as a result of thirst? Okay, so only the hypothalamic hormone must be mentioned, which is called vasopressin, which is synthesized in the hypothalamus and secreted from the posterior pituitary gland, and actually it is stimulating water reabsorption from the kidneys, therefore more concentrated, less urine will be formed. The other hormone that we discussed during the lesson, it is not a hypothalamic hormone, but it is produced in the adrenal cortex. It was called aldosterone, but it is not us. Okay, so that's it for the first option, and let's look at the second optional task, which is about plant growth. Auxin is the title. Darwin was one of the first scientists to study the way plants respond to light. In his experiments, he covered the shoot tips, coleoptile, of young plantlets with dark caps and observed the effect. Based on his results, he arrived at the conclusion that the light was detected by the tip of the shoot, while the bending of the shoot towards the light occurred below the tip. So we can see the experimental setups. In here, the growth of the shoot in the dark is indicated by X. When it is illuminated from the right, we can see Y, and evenly dispersed lighting can be seen on Z. The possible results of Darwin's experiment: the small cylinders are the caps placed on the shoot tips. What did Darwin experience when he covered the shoot tip with a dark cap with the line shown on the plant from one side? When giving your answer, compare the rate of growth shown in the figure on the left. Write the letter of the correct drawing, A to E, in the box. Okay, so basically here everything is written down in the text. So if he covered the shoot tip, obviously light couldn't reach it, therefore no bending occurred because the tip was not stimulated. We can read it in the text as well, that is the part which is detecting the light. And also, if you remember what we have learned about auxin, it is produced in the apex of the plant, and if the light is coming from one side, actually auxins will be redistributed on the shaded side of the plant. Remember, we discussed what is happening with proton pumps, pH changes, and expansins. But the big message, basically, of this is that cell elongation will take place on the shaded side, and that will cause the plant to bend towards the light source. Turning back to the question, so when the tip was covered, actually light couldn't be detected, therefore no bending occurred. So out of these, B will be the good answer. Later it was proved that a chemical was formed in the shoot tip which caused the plant to bend towards the light in part of the stem below the tip. Árpád Pál cut the tip of the shoot, which was kept in the dark, and put the tip back on the cut end of the shoot asymmetrically. As a result, the shoot bent away from the side covered by the tip towards the side which was not covered by the tip. Based on these findings, he arrived at the conclusion that the growth regulator substance moved down the stem, and in response to light it accumulated on the shaded side, where it induced more vigorous growth. In one of his experiments, Fritz went, after removing the shoot tip, placed the jelly block containing the growth regulator in question on the cut surface in even lighting, in a way shown in the diagram. We can see here what happened. Based on your knowledge and the information given in the text, draw in the diagram the direction in which the bending occurred. So the hormones traveled down the chute in this direction, and therefore bending occurred in this way. Okay. The chemical in question was named auxin. Later, two hypotheses were put forward in an attempt to explain how the accumulation of auxin took place on the shaded side when unidirectional light was applied. First hypothesis, the auxin on the side exposed to light breaks down or loses its activity in other ways. Second hypothesis, the auxin in response to light moves towards the shaded side. In order to test this hypothesis, 4 experiments were carried out, and their results are shown in the figures A, B, C, and D. The glass plate applied in the experiments is impermeable for water-soluble particles. The number below the jelly blocks show the amount of auxin diffused into them. Okay, so we can see here the experimental setup. After studying the experiments, decide whether the following explanations, interpret experiment A, B, C, or D, are based on their results, which hypothesis can be proved or disproved. When there is a glass plate preventing the auxin from moving laterally, the auxin is equally distributed in the 2 sides of the jelly block when the tip is illuminated from one side. The letter of the experiment can be figured out easily, so basically we just have to observe these images, and this third exercise is describing us D, so that's gonna be the letter of the experiment. Conclusion, the result, disapproves, proves, or neither proves nor disapproves the first hypothesis, which is the auxin on the side exposed to light breaks down or loses its activity. And actually this experiment disapproves this statement, so A will come here. Okay, when there is no glass plate preventing the auxin from moving laterally, the auxin concentration is higher in the shaded side when the tip is illuminated from one side. The letter of the experiment, illumination, we only have 2 options which are illuminated. Obviously the other one will describe this 4th scenario, so C will be the good answer. And then conclusion, the result, disapproves, proves, or neither proves nor disapproves the second hypothesis, which is stating that auxin moves down the shaded side in response to light. And if you look at these numbers, you can say that it proves the second hypothesis, so B will come to this box. Moving on. In the dark, when there is a glass plate preventing the auxin from moving laterally, less auxin diffuses into the jelly block than when no glass plate is applied. The letter of the experiment, so in the dark there is a glass plate. This is going to be the good answer, then B. And let's see what is happening. So, okay. All right, and conclusion, the result, ABC, second hypothesis, which was again, auxin moves down the shaded side in response to light. This experimental setup was in darkness, so actually C will be the correct answer in here. Exercise 6, explain how the plant benefits from its ability to bend towards the light. Very easy question. We know that plants can get more light, therefore more efficient photosynthesis could take place. Name another process in which auxin plays an important role. The most obvious answer is positive geotrophism of the roots, but if you check the key, you can also see that hydrotrophism would be accepted, apical dominance, elongation of cells, and so many alternative answers would be accepted. And we arrive to the last task of this high-level paper, dividing plant cells, an essay type question. And as we have done previously, a separate slide will be donated to this topic. And as we have done previously, a separate slide will be donated to this question. So, in your essay, discuss the typical locations and functions of the cell divisions in angiosperms. Include the following points: the different locations of mitotically dividing tissues, somatic cells, and their functions, and the location of meiosis, and the further division of the cells produced by meiosis until fertilization takes place. So let's start off by talking about the first point. We know that undifferentiated meristems of the plants are responsible for the indeterminate growth. Longitudinal growth is done by shoot apical meristems, root apical meristems, and the intercalary meristems, while the lateral growth is done by cambium. And that's what I wrote down in here. Moving on to the second point, and in order to elaborate this topic, I would like to show you a little picture which is showing us basically everything that is taking place in the ovary and in the anther, because these are the stages that we need to write down until fertilization, so these stages. Let's start with the female part. Ovule formation is taking place in the ovary, where one diploid macrospore mother cell divides by meiosis. As a result of this cell division, 4 haploid cells are produced. Out of these cells, one turns into the megaspore, and the rest will degenerate and die. The megaspore then divides by mitosis 3 times, which will result 8 haploid nuclei in a common membrane. And the embryo sac, which is the female gametophyte, is going to have 3 antipodials, 2 synergids, 1 egg cell, and 1 diploid central cell. So 2 nuclei fused together here, and the diploid central cell was produced. So let's see how does it look like if we write it down in a nice way. Ovule formation happens in the ovary of the female flowers and in the female part of the bisexual flowers. First, the diploid megaspore mother cell divides by meiosis, and out of the 4 cells, only 1 turns into megaspore. The rest will degenerate and die. This megaspore undergoes 3 mitoses to produce the embryo sac cells. Antipodials, synergids, egg cell, and diploid polar nuclei can be found in it. Embryo sac is the female gametophyte. Okay, let's move on to the male part. So meiosis happens in the pollen sacs of the anther, here. And not surprisingly, again, we need to start the process with the diploid cell, which is, in this case, is called diploid microspore mother cell. And this cell will meiotically divide, and as a result of this division, haploid microspores are produced. These microspores will develop into pollen grains, which are technically the male gametophytes. And pollen grains have 2 cells, a tube cell and a generative cell. We know that the generative cell, after pollination, will divide into 2 sperm cells. And those 2 sperm cells will do actually double fertilization, but that's something we don't need to elaborate in this exercise. So sadly, we need to stop in here, but I know all of you guys know double fertilization very well. So maybe in the future, in another exercise, you will have a chance to write down the process. So let's see how actually all of these can be written down. In the male flowers, meiosis takes place. In the pollen sacs of the anther, diploid microspore mother cell meiotically divides, produces haploid microspores. These develop into pollen grains. Inside the pollen grains, there are 2 cells, tube cell and the generative cell, which will later on divide into 2 sperm cells. So that was it for this high-level paper. I hope you found it useful. Should you have any questions, you know where to find me. And I wish you success in your preparations and the best of good luck for the final examination. Bye-bye.